Hello everyone, welcome back. Today we are going to learn about the bacterial cell structure. So, first we have to understand about the morphology and internal structure of the bacteria, which will be useful for us in classifying the bacteria and also to understand various physiological processes that are going to take place inside the bacteria. Bacterial size generally varies from 0.5 to 1 micrometer in diameter, where bacteria used to have higher surface area to volume ratio, which is favoring high rate of growth and metabolism. So there is no circulatory mechanism what we found in eukaryotes that is absent in bacteria. Because of this higher surface to volume ratio, these nutrients directly absorbed into the bacteria. Coming to the bacterial cell structure, bacteria is consisting of the various structures, external or present on externally or internally, and let us see each one by one. Here if you see the bacterial cell structure, here external structures like we can found capsule, cell wall and some flagella and pili, whereas internal structures include gel-like matrix, that is a cytoplasm, and some inclusion bodies, and here we can see nucleoid and plasmid DNA, and some in here mesosomes. In this picture we can see these are the foldings of the cytoplasmic membrane, and let us discuss each one by one very clearly. Coming to the cell wall, bacterial cell wall is very rigid, and because of this rigid nature it will give particular shape to the bacteria, that is like cocci and bacilli type of bacteria, and this gives cell wall give mainly protection from the osmotic stress conditions and harsh environments. Most of the bacteria, they retain their original cells even if we are subjecting to very high pressure or some severe physical conditions. The cell wall of the bacteria counts for 10 to 40% of the on dry weight basis, and this cell wall we can broken by using sonic or ultrasonic treatments, or if you are subjecting to the cells to extremely high pressure and sudden release of this high pressure, through these treatments we can break the bacterial cell wall. And here the cell wall composition is different in case of eubacteria and archaebacteria. Generally bacteria are divided into two categories based on evolution and genetic relatedness, that is, one is eubacteria and second one is archaebacteria. In both this type of bacteria, cell wall composition is different, whereas in eubacteria cell wall is made up of peptidoglycan. That peptidoglycan is basically a polymer of different amino acids like N-acetyl glycosamine, N-acetylmuramic acid, L-alanine, D-alanine, D-glutamate and diamino acid. This peptidoglycan is present only in prokaryotes but not in eukaryotes. Whereas in case of archaebacteria, this cell wall is generally made up of proteins, glycoproteins and polysaccharides. So main cell wall function is to protect the bacteria from harsh environments, and it give particular shape to the bacteria. And here cell wall is made up of peptidoglycans in case of eubacteria, whereas archaebacteria is made up of proteins, glycoproteins and polysaccharides. Coming to the one important treatment, that is technique to differentiate the bacteria into a positive and negative, that is Gram staining technique. This technique was developed by Christian Gram during the year 1884. This here we can use chemicals to differentiate, that is crystal violet chemical to differentiate the bacteria. So these gram positive and gram negative bacteria, both types are present in eubacteria and archaebacteria. If the bacteria appear deep violet in color after gram staining, then we can call it as a gram positive bacteria, whereas if bacteria appear red in color, then we can call it as a gram negative bacteria. Generally gram staining is applicable only for the bacteria but not to the other microorganisms like fungi. So coming to the second structure, so first one is the cell wall and second one is the flagellum. This flagellum is mainly useful for the motility of the bacterium. So this flagella is which is hair-like helical appendages protrude through the cell wall and it is responsible for the swimming mobility. That is useful for the mobility of this bacteria from one place to the another place, and it grows at the tip unlike hair which grows at the bottom. Hair used to be grow at the bottom of the bacteria, whereas this flagella used to be present at the tip of the bacterial cell. This flagellum is composed of the mainly three parts. One is the basal part, that is basal body, which is associated with cytoplasmic membrane and cell wall. In this picture you can see the different parts of this flagellum. The basal body, which is also known as motor device, especially this is responsible for providing some energy for the movement of the filament region. Then second portion is the short hook. Here this is the short hook, and third one is the helical filament, which is usually several times more longer than the bacterial cell. This hook and the filament, this is made up of protein, which is protein, whereas the basal body composition is not clear, and the protein present in this filament region generally known as flagellin. And based on flagellar arrangement, bacteria can be divided into different categories. Like if the flagella is absent in the bacteria, then we can call it as a atrichous bacteria. If the bacteria consisting of the single polar flagella, we can call it as a monotrichous. And if the bacteria is having a cluster of polar flagella, it is known as lophotrichous bacteria. And if the flagella is present either in a single or in clusters on both sides, then we used to call it as a amphitrichous bacteria. And another bacteria is the peritrichous. Peritrichous means the name itself indicating. Peri means that it is present throughout the bacterial cell. That is, if the flagella present throughout the bacterial cell, then it is known as peritrichous flagella. So how this bacteria will move with the help of this flagella means bacteria propel themselves by rotating this helical flagella. So without external, some bacteria we can see without flagella they show mobility, that is spirochetes. These are the example which exhibit swimming mobility due to the presence of endoflagella. Endoflagella means flagella which present below the outer cell envelope. So this endoflagella we can observe in spirochetes. Okay. Then in case of some other bacteria like myxobacteria, they will exhibit gliding mobility, that is gliding mobility when they will come across the solid surfaces. So endoflagella we see in spirochetes, and gliding mobility we can observe in the myxobacteria. And remaining bacteria, they will exhibit movement with the help of flagella, which are different types like monotrichous, lophotrichous, amphitrichous and peritrichous flagella. And we can observe the different type of movements with respect to the some external environmental stimuli. Such type of movements in the bacteria we used to call it as a tactic movements. That is, motile bacteria are able to change their movement in response to environmental stimuli. So that is known as tactic movement. There are two types of movements. One is chemotactic movement and phototrophic movement. In chemotactic movement, if the bacteria move towards the chemical, that is known as positive chemotaxis. If the bacteria is swimming away from that chemical substances, then it is known as negative chemotaxis. And in case of phototrophic bacteria, if the bacteria is exhibiting positive phototaxis, that is moving towards increasing light intensities, then we can call it as a positive phototaxis. And if the bacteria is repelled by decreasing light intensities, then negative phototaxis. That is, they are moving away from the high intensities of light, then it is known as negative phototactic movement. So another structure is the pili and fimbriae. These are structures present throughout the bacterial cell. which are thinner, shorter and more numerous than flagella, and their function is not in the mobility, but they have different functions. Like if you will take F pilus, that is X pilus, which serves as a entry point for the movement of the genetic material during the bacterial mating. And some pili play major role in the human infection for attachment of this bacteria to the human cells. This pili also play very important role. Understand? Then coming to the another structure, that is capsule. Capsule present outside of the bacterial cell. So the capsule is made up of mainly organic exopolymers that form the envelope outside the cell wall. So if we will see this layer by light microscopy using special staining methods, then we can call it as a capsule. If you will observe under light microscopy this structure, then we can call it as a capsule. And capsule, here we will use two terms, like one is microcapsule and another one is the slime. So where we will use this microcapsule term means if the capsule is very thin to be seen under the light microscopy, then we can call it as a microcapsule. That is the name itself indicating that micro means very thin in nature. And we can call it as a slime if the layer is abundant and many cells are embedded in a common matrix that is very thick in nature, then we can call it as a slime. So most of these pathogenic bacteria, they used to produce either capsule or slime. So in capsule we will use two terms, microcapsule and slime. Then function of the capsule, how this capsule play an important role, because it will block mainly attachment of the bacteriophages. That is, bacteriophages are the viruses which will infect the bacteria. So when these bacteriophages come in contact with the bacteria, capsule prevent the attachment of these bacteriophages to the bacteria. And these capsules are antiphagocytic in nature, and they also provide protection against temporary drying, because these water molecules, they used to attach to this capsule, and this capsule will prevent the drying of this bacteria. But this is a temporary. And they may promote attachment of bacteria to the surfaces already during the infection process, either in human infections or pathogenic this plant infections. These capsule promote the attachment of bacteria to the surfaces. And after capsule, we can see sheath-like structures in some bacteria, especially under aquatic environments. These sheath-like structures we can see for the bacteria. What is this sheath? Sheath is a hollow tube formed in some species of bacteria to enclose a chains or trichomes of bacterial cells. Here you can see the sheath-like structure where bacterial cells are enclosed, and this sheath is commonly found in freshwater and marine environments. And coming to the internal structures, so up to now we studied about the external structures present on the bacterial cell. So inside the bacterial cell there are some other structures, so let us discuss one by one. First one is the cytoplasmic membrane. Cytoplasmic membrane is about 7.5 nanometer in thickness, and it is present immediately beneath the cell wall. This cytoplasmic membrane is mainly composed of the phospholipids 20 to 30%, but major portion is the proteins, that is 60 to 70%. And the cytoplasmic membrane is very important membrane because it contains various enzymes. That enzymes are involved in the respiratory metabolism, that is respiratory mechanism of this bacteria, and in synthesis of this capsule, and also cell wall components. And the cytoplasmic membrane is the site of generation for the proton motive force and ATP generation and certain nutrient transport systems and movement of the flagella. Because this cytoplasmic membrane, it will generate some ATP which will be also helpful for the movement of this flagella. And if we are damaging this cytoplasmic membrane, generally bacteria used to die. And we can found on some other structures like protoplast and spheroplast. Protoplasm is nothing but the space between the cell wall and the cytoplasmic membrane. That space is known as protoplasmic membrane or protoplasm, we can call it as. And protoplast, it is the portion of bacterial cell consisting of the cytoplasmic membrane and cell material bound by it. And here cytoplasm in case of gram-positive bacteria. If you are treating the cell with the enzyme, that is bacterial cell, if you are treating the enzyme with a lysozyme, then it selectively dissolves the cell wall, but it does not dissolve the cytoplasmic membrane. Thereby we can obtain this protoplasm. Or if you are treating the bacterial cell with antibiotic penicillin also, we can get this protoplasm. And spiroplast, this is the protoplast surrounded by outer membrane of the cell wall. In case if you will take the gram-negative bacteria, only if you are treating with any antibiotic, we can remove the peptidoglycan layer of the cell wall, but this outer membrane of cell wall, it will used to be remain, so that we can obtain the spiroplast. And mesosomes are also we can observe in the cytoplasmic membrane. These are the foldings of the cytoplasmic membranes. Those structures we used to call as a mesosomes. So let us see what are the mesosomes. In many bacteria, especially gram-positive bacteria, the cytoplasmic membrane which appears to be enfolded at more than one point, and these foldings, that is enfoldings of the cytoplasmic membranes, are known as mesosomes. And they what is the role of these mesosomes means they are thought to be involved in DNA replication, cell division, and export of extracellular enzymes. And coming to the inside, that is cytoplasm, gel-like matrix, what we observed inside the cytoplasmic membrane, that is known as the cytoplasm. This cytoplasm consisting of the various components like nucleus. Nucleus used to be present without any nuclear membrane, and ribosomes, proteins, and any other water soluble components, and some reserve materials. So along with this, some extra chromosomal DNA, that is known as plasmid DNA, also present in the bacteria. This plasmid DNA is responsible for imparting antibiotic resistance to this bacterial cell. And bacterial chromosome, here nucleus is not enclosed in a definite membrane structures what we found in the eukaryotes. Here there is no particular nuclear membrane. So hence this nuclear material we used to call it here as a nucleoid or chromatin body or nuclear equivalent. And this genetic material is a single circular double stranded DNA molecule where it contains almost all the genes responsible for various functions of this bacteria. Here in this picture you can see this is the nucleoid and these are the plasmids. And ribosomes, here ribosomes of the prokaryotes, generally 70S type, which are consisting of the two subunits, that is 50S and 30S subunits. And some ribosomes used to be free in the cytoplasm, and some ribosomes used to be attached to the cytoplasmic membrane. Mainly they are responsible for the protein synthesis. And beyond these structures we can observe also volatile granules and polybeta hydroxybutyrate and glycogen. These are some of the granules present in the cytoplasm of the bacteria. Along with this, and we can also observe the gas vesicles which are that present in the aquatic environmental bacteria. These are the different structures present in the bacteria externally, internally. And beyond this we have to study some spore-like structures which are produced in the bacteria. I think you heard about the exospores and endospores. Let us see what are those spores produced by the bacteria. And what is the spore? Spore is generally, it is a metabolically inactive structure, that is metabolically dormant form under produced under harsh conditions. So under appropriate conditions, what will happen? This spore undergo germination and they used to grow out to form a vegetative cell. And if the spores are produced within the cell, they are known as endospores, and if they are produced outside the bacterial cell, they are known as exospores. And endospores, generally how they look like, they are very thick walled and highly resistant and refractile bodies. And these endospores are produced by Bacillus, Clostridium and Sporosarcina. And these endospores are formed, these are produced inside the cell. So when they have to produce, they used to produce at the end of active growth or during stationary phase. Advantage of these endospores to the bacteria is they are extremely resistant to the desiccation and any staining technique and disinfecting chemicals and radiation and heat. exospores are formed external to the vegetative cell by method of budding, and we can see exospores in the methylocyanus, and these exospores are desiccation and heat resistant. And coming to the other terms, conidiospores and sporangiospores, where we can observe these terms in the actinomycetes, which form the branching hyphae. From this tip of hyphae, these sporangiospores or conidiospores used to produced. If these spores are produced in enclosed sac-like structure, that is sporangium, we can call it as a sporangiospores, and if they are produced freely, then we can call it as a conidiospores. So these spores can survive long periods of drying, but they do not have any high heat resistance as what we observed in case of endospores and exospores. And coming to the another structure, cyst. So cyst means the name itself, it is indicating that it is like a hard-like structure and it resembles the endospore, but structure and composition is different for both endospores and the cyst. Cysts are thick-walled and desiccation-resistant dormant forms that develop by differentiation of vegetative cells. We can observe the cyst formation in the azotobacter. So this is all about this bacterial cell structure and structures present externally and internally to the bacteria,